organ in the universe. It has consumed scientists and philosophers for millennia. As a neurobiologist, I've been trying to unlock its secrets, but it's really difficult to study a developing human brain. This challenge has taken me on a journey of discovery. The result will hopefully lead us a step closer to unraveling this greatest of mysteries. I first got interested in the human brain when I saw a neuron for the first time. When I was a child, I used to visit my dad at his laboratory. He invited me to look through a microscope at a neuron that they had growing in a dish. And I was completely struck by how beautiful and how complex the neural structure is. It was just amazing. There are 100 billion neurons in the human brain, which is equivalent to the number of stars that there are in our entire galaxy. The beauty of it still gets me every time I see a neuron. So the challenge has been that we haven't really been able to actually probe a developing human brain in utero. And so the only thing we really could probe was animal models. But of course, they have major differences to a human brain. We really wanted to find a way to be able to actually study human developing brain tissue. So working with Jürgen Knoebel, I started with skin cells, which can be programmed to something called pluripotent stem cells. And these can then generate any cell type of the human body. What we did is push them to start becoming brain-type tissue. I spent many hours in the lab working with different concentrations of nutrients and different vitamins to try to find just the right environment for the tissues. But then the real breakthrough came when we actually placed these tissues into a very dense protein gel that allowed them to self-organize and form the architecture of an actual developing human brain. Previously, others had generated individual brain regions in isolation. We were able to generate the first three-dimensional cerebral organoids that contained all the different brain regions. When I first saw these mini brains, I was really in awe. Although they don't have any sensation or any feeling, we now had this tool that we could use to look at all kinds of questions about a developing human brain. I think probably the biggest impact that this will have is in the area of neurological disorders like autism and schizophrenia. We can generate brain organoids from patient skin cells and start to understand what goes wrong in those disorders and what can we do to try to alleviate the symptoms. When I think back on that first time that I saw a neuron in a microscope, I would never have imagined that I would be able to contribute to human brain research in this way and bring about this technology that biologists can use to discover really exciting new things about the human brain.
The Genius